Hey guys, today we have our Kool-Aid analysis lab. So what I have prepared for us, I have made some concentrated solutions of Kool-Aid coming from the powder, and we're going to see how the powder compares to a pre-made Kool-Aid jammer. So when we are doing the analysis of this, we are going to use absorption spectra to tell us how much light is getting through our sample. So this connects to our idea of our absorbance versus our transmittance, and that connects to how we see colors and what light passes through them. So we have red solutions. I have cherry Kool-Aid here, and so we're going to expect to see peaks in the blue region of our graph, because when a substance is a certain color, it absorbs its opposite color, if you think about your primary color wheel. One of the main ingredients in our Kool-Aid today is a red food dye. It's called Red 40, and I actually have a sample of this for us to get a baseline to see where our absorption spectras fall. So I'm going to go ahead and insert this into my SpectroViz. Kind of the setup of this is that there is a light, a white light, that goes through a prism, and that's going to separate that white light out into its different wavelength colors. And so as it does that, then we're going to be able to see what type of light absorbs most through the peak on our graph. There's a detector on the other side of the light, and so that's where we get our data from. So I'm going to go ahead and stick this red 40 in there, start my data collection. Once I make this a little better, as you can see, in our blue kind of green region, we get a high peak. So I'm going to go ahead and collect this data. That'll be kind of our baseline, and I'll go through and label it later. So now I have different concentrations. My first concentration will be 2 molar. This is where your data collection starts. So I'm going to insert that into my SpectroViz and again collect the line of it. Okay. And now you can start to see where we are getting that kind of overlap between our dye and our Kool-Aid itself. Collect this one. Next up is our 1 molar. As you can see, it still shares that similar peak. I'm going to collect this. We'll have our 0.5 molar. So each sample that I run gets less and less concentrated. And lastly, our 0.25 molar. There's our last. So these are our known concentration solutions. Our job is to figure out what concentration our jammer and where it falls within our range. You'll be able to eventually use your data in order to figure this out, but we'll go ahead and get it on our graph so that we have an idea of where it is. So that is ultimately our last data set for this. Okay, so now I'm gonna go ahead and annotate this. Should be good. All right, so now that we have run all of our samples and we have a good graph of each of them and kind of where they are with respect to each other, we're gonna take a moment and write down our absorbances for each of our peaks. And that's gonna basically tell us what wavelength had the highest absorption within that sample. So as we look at our 2 molar, we're going to be looking at the blue kind of text here. So you can go ahead and write down 1.702 for your absorbance on the data table on your lab handout. We'll hop down to our jammer. Our jammer is the green that we see here on the graph, and that's going to be 1.504. The 1 molar is going to be right under that, and that is 1.499. Lastly, or I guess not lastly, then we have our 0.5 molar. That's going to be in our orange text here on the screen. That's 1.383. And then way down here at our 0.25 molar, we have an absorbance value of 0.531. So I'm hoping that you guys are able to pick up on the trend of what concentration does for our absorbance. The less amount of color that we have in our solution, the lower the absorbance is going to be, and the more transmittance we're going to have. So at this point, you have all the data you need. You can return back to your lab handout and follow the directions to graph the concentration versus absorbance, and then start to predict what the concentration of our Kool-Aid jammer will be.